Welcome to the July 2023 Partners Meeting of the CCT. As many by now know, our focus this year is to help stimulate coral rescue programs in as many jurisdictions as possible, to collectively give us a chance of preserving some genotypes representative of the Caribbean's unique endemic biodiversity in those species of corals that are most at risk of local ecological extinction due to SCTLD. This is the third of our bi-monthly instructional webinars in pursuit of that objective. We have a full agenda today, so let's get this started, especially after this rather chaotic beginning. Dr. Anastasia Banaszak, who's a distinguished researcher at the UNAM lab in Puerto Morelos, Mexico, already gifted us with a riveting introduction to monitoring coral spawning in May. I am so thrilled that she was willing to return to visit with us today. She will first explain how to build spawn collecting nets for those of you who feel ready to experimentally practice artificial fertilization, even if embryos are then returned to the sea to fend for themselves. She will also discuss with you what has been learned so far this year about the spawning behavior of the brain coral DLAB and give us her 2023 predictions for some of the other species that are expected to spawn during August to October. And here now is Anya, and I will stop screen sharing and let her begin.
Hi, everyone. I hope you're seeing my screen.
Yes, we are.
Okay, perfect. Well, thank you for joining us today, and thank you for the invitation to come back. I'm very happy to be here and to share this information with you. Let's see. Okay, so first we'll be talking about fabrication of collecting nets. In our lab, we based our design on a collecting net that Alina Schmant brought to us about 12 or 13 years ago now. And we have modified it a little bit and we think it works perfectly. So we'll share with you just how to make these. So this is the net. The basics are that it has obviously netting material. We use a netting material that we can find in just regular fabric shops here because Mitex netting is so expensive. It's basically what's used as mosquito netting for people who use hammocks. So it tends to be available in fabric shops. But if you can't get it, let us know and we can always send you some material because it's so cheap to buy here. And basically at the bottom there are our weights that you can see here, and these are basically fishing weights, again usually available in fishing stores. They're kind of like round cannonballs shape and then have a groove in them so that they can be sort of fitted onto the netting. And then basically at the top of the netting is actually we use two funnels, one nested on top of another, and basically a connector, and then a rope with a float, and then the collecting cup. Here it's about a 50 mil or 100 mil collecting cup, but we now use 50 mil tubes, and basically for our collections, as we're trying to collect less from each colony, but from more colonies. So this is basically close up of the two funnels and then the rope that leads up to the float. And then we basically then always put tags onto our collecting cups so that we can tag, put the number of which colony and what time it spawned. So in order to fabricate it, we basically make a mold just using sort of brown paper, and the instructions are given in an instruction manual that has the guidelines. You can copy it from this site. I can always put the link into the into the chat later on. And there's also a video available that takes you step by step. The manual has photos, and then the video obviously has sort of a step-by-step instructions as well as to how to do it. And so if you would like a copy of that video, you can just write to me and I'll send you the link. And these are basically the fishing weights that we use that have this groove. And like I said, we just give step-by-step instructions as to how to fabricate the net. And I think it's also important to talk about net placement and just sort of the idea behind it. So generally, once the dive starts, we put all of the collecting nets into a staging area. And then from there, they're basically distributed out. The very nice thing about this design is that if you put the net over a colony that you think might be spawning, but then see a colony nearby that is spawning and you want to change the net, it's really quick. It's a quick operation just to take it off the colony that is not spawning and put it onto a colony that is spawning. So in terms of correct placement, if you see these nets are not sort of spread out completely, we basically keep quite a tight angle on these. You can see, especially here, that the net is sitting upright but very, very low angle. And the reason is that if we spread out the net too much, what's going to happen is that the gamete bundles are going to escape through the net. Okay, they're going to get caught up in the net, let's say here. And then because of the wave action, the gamete bundles will break up and then the gametes will be lost. So basically, what you're aiming for is to collect almost directly under the funnels. And also one really important point is to, in your collecting cups or collecting tubes, make sure that you remove all the bubbles, because if you don't, the gamete bundles can start breaking up with the wave action and the bubbles present there. Also, you don't want to have an excess of nets on a single colony. It's really better to limit to having one net per colony, because if you saturate your fertilization mix with a lot of gamete bundles from one particular colony, you'll likely have very low fertilization. So, sorry, this is a bit out of place, but basically this is the correct net placement. You do not want to put the net spread out like this. Of course, this is an exaggerated photo, but it's just to make the point that the gamete bundles basically don't know that they're supposed to go up into the funnel and into your collecting cup, right? So because they have a lot of lipids and have this floatability to them, it means that basically when they float up, instead of floating up into the funnel, they're going to float up onto the actual material. And because, you know, it's slightly open, basically all of those bundles will just basically break through the net with the wave action. So during spawning monitoring, what you want to do is to review the nets periodically for bubbles. And if you do find bubbles but no spawn yet, just remove the cup or the tube, turn it upside down and let the bubbles leave. Just shake the collecting cup or tube and the bubbles should come out. Also, try to avoid putting light directly on the nets. I think we spoke about this last time, but if you try to avoid any direct light on the colonies and on the collecting nets, because it attracts predators. And what can happen is that a predator will swim up into the collecting cup and eat all of the delicious gamete bundles. Also, when you're putting the lid onto the tube or collecting cup, make sure you keep the collecting cup and tube upside down, and make sure to place the lid correctly and maintain the collecting cup lid side down. And if you pass it to somebody in the boat, make sure you tell them to keep the collecting cup upside down as well. And only turn it right side up once you're ready to do the fertilization step. Ensure that you write down the time of setting, if you see it, and of spawning, preferably the start and end of spawning. And we spoke about logging the data and maintaining your data sheets both underwater and on your computer. We spoke about that last time. So here are some examples, for example, with Acropora, just for how a setting looks. So you can see these are bundles in the mouths of the colony. And a few important points, sometimes setting and spawning will occur underneath the colony. But also what you will note is that, for example, in this colony, and this is very common, that the edges, the growing edges of the colony will not have setting or spawning. It's usually the branches further inside the colony. And I guess just because they're the growing tips, they have energy either for growing or for reproducing. And so these are the growing edge that don't produce gamete bundles. You can also see that here, right, where you can see the spawning as well inside the colony. So that's another reason to not have to put the net and spread it all out to cover the branches. So that basically finishes that section. I don't know if there are any questions or comments on that. I don't mind at all being interrupted while I give the talk. I'd rather answer any concerns or questions as we go. Are there any questions in the chat by chance? If anybody's looking, I can't see the chat.
There are no questions yet, but if anyone has questions, you can ask it now or write them in the chat box.
Okay, perfect. Thank you. So then we're just going to move topics to the spawning update on DLAB, because it has started to spawn in some areas of the Caribbean. Sorry, just kidding. So in terms of the predictions that we showed last time, we showed that from June to October, the most likely months for spawning are June to September, the highest probability of spawning. And that the most likely dates were the 13th, 14th, and 15th of the months, with the nights after full moon being the 10th, 11th, and 12th nights. Whereas the 9th and the 13th night after full moon, there's probable spawning, but not as likely as when the 10th, 11th, and 12th night after full moon. The predicted times were from 30 to 55 minutes before sunset, and spawning from 10 to 100 minutes before sunset. And those are the citations from where we took this from the spawning prediction calendars that at least I have access to. So in reality, what happened, at least up in June, was that in Cuba, the Acuario Nacional de Cuba from Universidad de Havana, they went out for spawning and found that on the 12th, 13th, and 14th of June, 3, 6, and 6 of 79 colonies spawned. So, you know, it's a small number, but still it's good enough for assisted fertilization. But definitely it's not necessarily the highest level of spawning so far. So that occurred 9 to 11 nights after full moon, so within our predictions, from 1853 to 1935 hours. In Dominica, Nature Island Dive went out and found spawning on the 14th of June, where 3 of 21 colonies spawned, and that's the 11th night after full moon. And for the US, the Florida Aquarium Center for Conservation found that they had 9 and 5 of 22 colonies spawn on the 13th and 14th at 1812 to 1845, which are 10 and 11 nights after full moon. And in Mexico, we did go out in June just in case, but we did not observe any spawning, which is our prediction. Our prediction is that Diploria labyrinthiformis from all the previous years that we have been following doesn't spawn until July and August, but we went out just in case. So are there any more DLAB reports for June? Has anybody else gone out and could let us know? Alice, if you could. Hi.
Hey, yeah, so I did go out in June. I didn't go out on the 12th. I went on the 13th, 14th, 15th, and 16th. Unfortunately, we did not see anything. But we did in May.
Okay.
So I will be going out again this week. We'll see what
Okay,
happens.
wonderful. Good luck. And
Sorry, I
so
haven't filled in the forms, Anya. I will
yeah,
do it.
no worries. Thank you so much. I really appreciate that. So sorry, where are you diving? Sorry, I can't remember.
Turks and Caicos.
Oh, lovely. Okay, wonderful. Thank
Now,
you. very
where in
much.
Caicos, Alize?
So this was off of Providenciales, on the west side.
Okay, wonderful. I think Gabriela also had her hand up.
Yeah, we also observed June in Roatán and Guanaja, and our colleagues in Utila also observed it. I'll have to double check the day exactly. I was a bit busy with the training, but we saw about 8 of the 20-something that we were monitoring. And in Guanaja, they saw even 30 out of the, I don't even know how many they were monitoring because they're still on the epidemic phase of SETLD. So they have quite a few DLABs. So it was really beautiful. For those that observed it, I was in Roatán, but I will send your form to the Roatán Marine Park.
Oh, wonderful. Thank you very much. Yeah, that would be great to have all those data. They're so valuable.
Definitely.
I see that Leslie Henderson has some observations. Do you want to tell us, Leslie?
Hi, can you hear me?
Yes.
Hi, yeah, I just wanted to say that we did spawning observations in May and had quite success. I actually wasn't on those dives, so I don't know the numbers. But then in June, only 1 out of 10 spawned all week. We monitored like 5 days, and it's just a single coral. So I'm not in charge of that project, but I'm assuming that the folks who are submitting data somewhere. But if there's somewhere that I should be telling them to submit it, let me know, and I can make sure that's happening.
Yeah, that would be lovely. Yes, we gave that information in the last talk that I gave, but we can certainly send you the links or copies of the spawning monitoring data sheets. That would be really wonderful. And sorry, where were you diving? I didn't quite catch that.
Thank
The US
you.
Virgin Islands. Yeah.
Okay.
Simon, would you like to say something about what happened in Dominica, even though you were away? Is Simon on the call?
I'm not sure that he's on the call. But yeah.
Oh. Well, I guess not. Oops.
Okay, anybody else? Okay, so basically, I think we can pretty much sort of safely say that at least in terms of the Caribbean, in general terms, the 12th night after full moon is probably not, it's less likely to have spawning than the 10th and 11th night after full moon. And so, for example, here in Mexico, we'll be going out on the 13th and 14th of this month, because those are the nights that we've had, or at least the 10th and 11th night after full moon are the nights that we've seen spawning in the past. So it'd be really interesting to get data from other people, and so that we can start to build maps and see whether this is sort of a regional or localized thing that they only spawn on the 10th and 11th night, or if there are more nights. But the whole point of doing this is that as we have more information, we can make our spawning monitoring dives more efficient, right? Because so in our case, instead of going out all 5 nights, we know that we only need to go out on 2 nights. And in fact, I do suggest also that you look at it reef by reef, because we see differences even in the same species, but populations on different reefs will have slightly different spawning behavior, where one set will go off one night and another set will go off on a different night. So certainly it's really good idea to gather any spawning observations that you have for your area past and whatever you're doing now. It's highly suggested. Whether you send it to us or not is, a decision for your organization, but I highly recommend doing that for your organization.
Perhaps we should ask at this moment, how many people on the call will be looking again this July, or maybe looking for the first time this July, to see if they can monitor spawning of DLAB at their location. And if the answer is yes, why don't you write it in the chat? Oh, and Gabby has a question.
No, I think they're signaling that they are going to go out for spawning this month. Is that right?
I had a question for you, Dr.
Oh.
Anastasia. So we are still, this is our 3rd year, I think, validating our monitoring, and we have not observed DLAB in July, but do you think it's still worth going again? Last year we didn't observe it. Should we still go out in July again?
Well, okay. I think it depends on your resources. If you have limited resources and for 3 years in a row have not seen spawning in July, then perhaps what you have is an early, sort of earlier in the year spawners and later in the year spawners. Have you gone out in months around July and seen spawning, or?
We went last year, June, we observed spawning. July, we didn't observe spawning. So this actually would be our 2nd year. We've done 3 years for other species, but this would be our 2nd year for DLAB. So since I've seen that your peak is in July, I was just curious if you would recommend, going again this July just to be like 100% sure, or going days before the predicted window.
I would go again, depends on your resources. If you're tight for resources, I would go, but like on the 10th and 11th night. And because as you can see those are the nights that are most likely for spawning to happen, at least just looking around the Caribbean. If you're not resources limited and you can go out, let's say on the 12th to the 15th or even the 16th, then I would go for it. Yeah, just based on our data, where we do get spawning in July and August.
Okay, thank you.
And we sometimes get a dribble in September too. So, yeah. Any other questions? So let's move on to talk about other species spawning predictions. So for Orbicella fabulata, what I did was take the 4 calendars that I had access to from Curacao, from the people at Camavi, from ours in Mexico, from the Bahamas with the Perry Institute, and from the Dominican Republic with Fundamar. And so basically what I have done is put all the predictions for each month. So for example, in Curacao, there's sort of a likelihood of spawning of Orbicella fabulata in July. In Mexico, we haven't ever seen Orbicella fabulata spawn in July, but we have seen it spawn in August, September, and October. And And in the Bahamas also, there's a higher likelihood in August, whereas in Curacao, there isn't such a high likelihood in August. But as you move into September, there's a much higher likelihood, and in October as well. In the case of Mexico, we saw it once in October when I think it was the 1st and 2nd of October. So I haven't put it here, but basically it was on the 5th night after full moon. But what you can see is the most likely nights for spawning for Orbicella fabulata on the 6th and 7th night after full moon. So again, if you are resource limited, then my suggestion would be to go out on those 2 nights in August, September, and maybe October. And spawning in general runs from 185 to 255 minutes after sunset. In the Dominican Republic, oh, my screen is from 130 to 150, and from 220 to, I think it's 250 minutes or something like it's. I'm getting covered by a bar and I can't see I can't remember the data point, but after sunset. So what's interesting is they have an early spawning group and a late spawning group in the Dominican Republic. Here are the data for Orbicella annularis. It's sort of similar, a little bit more widespread times, especially in September. But basically, again, the 6th and 7th nights are the most likely. Although if you're close to the Dominican Republic or Mexico, maybe go out on the 4th and or 5th night after full moon. And spawning runs from 185 to 250 minutes or so after sunset. With Pseudodiploria strigosa, it spawns basically 6 to 8 nights after full moon in August, September, and October. Again, there appears to be an early group in Curacao from 35 to 100 minutes after sunset. And then there's a later group from 140 to 145 minutes in the Dominican Republic, from 180 to 270 minutes after sunset in Mexico, and from 205 to 270 minutes after sunset in Curacao. Here's a video of strigosa spawning taken by Raul Tecolco. He's a technician from CICO International working in my lab. And I just thought it'd be nice to have a break from my voice and see spawning. Okay. Oh, you go. And last but not least, Meandrina meandrites. This is a gonochoric species, so it has separate male and female colonies. It does lack a clearly defined spawning window, so it is a bit of a challenge working with this species. In general, it could spawn anywhere from 5 to 31 nights after full moon in September, October, and November, at 10 to 70 minutes after sunset. So I've basically summarized data from the Curacao prediction calendar, as well as you can check out O'Neill et al. 2021 in Frontiers in Marine Science, where they talk about this species and about Dendrogyra cylindris as well. And the photo is of a Meandrina meandrites male colony spawning. So are there any questions? That's basically the end of my slides.
Well, before you get any serious questions, I'd just like to ask you, how was that video taken inside the spawning without releasing the spawn and able to rotate around during, you know, instead of being just in one fixed position?
It was a very able diver, Raul, basically put a GoPro camera sort of under the net very discreetly, and I think it gave an impression of going around, but in fact it's just sort of the wave movement or the
rocked
movement
it.
of the current. Yeah. But I think it's a really neat video to see.
Well, I know the video is great, and especially because it really exemplifies what you were talking about, that the spawn may be, floating, but it doesn't know to go straight up, and the wave action can really knock it around. So if you don't have your net set correctly, I could see it easily, getting affected by that. So that was really great video.
Yeah, exactly. Simon, you have a question?
Yes, good afternoon. And my question, fairly predictably, is to do with meandrina. We have quite a bit of meandrina here on coral trees, and some that is still on the reef, but not much left on the reef. I'm wondering if there are any techniques. How can we narrow this down when I see you have, with our limited resources, you're looking at 5 to 31 days after full moon. Everything we have is in situ. Are there any techniques? Are there any methods that anybody is aware of where we could try to monitor this and get a better prediction or results?
Yes, actually, that's why we included the meandrina, knowing that you would be interested in it. I think it's really hard to follow this species in situ. If there's any possibility of having an aquarium set up, or even buckets with air pumps and bringing some colonies in, and having them in buckets and follow spawning, and have several, or in a large container with air pumps, and if possible having seawater at the temperature that they're found on the reef, because unfortunately, of course, if you bring seawater onto land, it will start to heat up. So yeah, I think that's the best possibility. If you know of anybody at all who's interested in aquaria or can run aquaria that could help you with that, I think that would be really great. Or perhaps have sort of maybe try to bring some of these meandrites together. I guess you mentioned they were on a tree, so they're pretty close together. And I guess it just would be really hard to go out all those days in a row. But if they could be brought into an aquarium tank, so I guess somebody just mentioned turtle tanks. If that's a possibility, I think that's your best bet.
Okay, thanks. Unfortunately, our turtle tanks are now taken down because we ran 2 projects back to back and released 12 turtles, but now they're shut down. So my next question is, if I could build an aquarium or find somebody to assist me, and I have a couple of people in mind, is there such a thing as a very, very small, sort of affordable heat pump or way to keep this? My biggest concern is not only would the temperature warm up, but also, overnight you'd have a fluctuation in temperature that might be challenging for meandrina to survive. So we could change the water every morning for a month. That's the kind of resource we could do. But is there any affordable little heat pumps or whatever, water chillers or?
Portable ones, you mean ones that run on gasoline or something rather than on electricity? Or
No. Electrical. The trouble is the heat pump I have costs me $2,000 US a month to run for the turtle tank. So I don't want to run that for a 200-gallon aquaria. Is there such a thing as a small heat regulator that we could use?
Yes, there are chillers and there are different sizes, just like air conditioners, because basically a chiller is sort of like an air conditioner, right? Just running water through it. So yeah, you can get different sizes of chillers, and we can send some information to you if you like. But I think basically it depends on what you would be working. Mhm.
I see Alize just said something there. If I could keep an air-conditioned small room stable at
As
an
well.
air temperature of 86 degrees, that would work as well.
That's
Yeah.
what we do, Simon. Yeah.
Mhm.
Yeah, I saw your comment there, Alize. Okay, thanks
Yeah,
a
so
lot.
we've got it set at 25 Celsius, and I mean, I'm looking to replace it with solar AC because our power bills are expensive, but they're not $2,000 a month. But if you have a small room that has natural light and you can just keep it HVAC'd, then it seems to be all right.
Yeah, we have the first phase of our like embryo and larval culture. We keep an air conditioner going. The thing is, because seawater will evaporate because of evaporative cooling, you need to keep the temperature higher than what you would want. So during culturing, we actually have our air conditioner running at 29 or 30 degrees. Because we want to keep the water temperature around 27 degrees. That's the optimal for embryo and larval culture, and possibly for maintaining the corals as well. Because at 25 degrees, maybe, I'm not sure what the water temperature would be, but it could maintain it around 24, I'm guessing, which is a little bit too low.
Simon had to leave to take care of something, but
Yeah.
thank you, Anya and Alize,
No worries.
for good comments. The one other thing, and I'll follow up with Simon on this, and I'm not sure if anyone else has tried it, but when I used to work with Harbor Branch and we worked in the deep sea, we had time-lapse cameras that we would leave at the bottom of the ocean that you can set up. So I'm not sure if that's a possibility for folks. I do not know how they cost, but I know the technology has advanced significantly over the past couple decades, but that might be an option, especially since he has them on trees, to attach some kind of small time-lapse camera and just kind of set it up so that they could observe it.
That's a fantastic idea. And then you could do all sorts of neat things of having it available to the public so that they can
Yes.
see.
And they do have them where you can have them attached to a cable, but they also have them where you can just set them down there and they run on
Right.
battery, and you would run ours for, several months at a time. So
Yeah, that's
just an
wonderful.
idea. I'll have to look into that.
Fantastic idea.
Yeah. I was going to add that Kimani Kitson-Walters was doing that with some GoPros off of St. Stacia last year during the DLAB potential spawning period. So we should ask him if they had any success. I'll
Superb.
add that to my agenda. So is anybody else planning to monitor any of the corals that spawn in August to October during any of the nights that Anya is suggesting, for any of the corals that she mentioned, or for any other corals either? Somebody might want to know if CNET, Copophilia natans, when it spawns, because there are some areas in which it's been very badly affected. And I see Steve Broadbelt came in, yes. Please say where you're planning, like in Cayman for Steve, thank you, and what species. And if
Yeah, we'll be going out in September, 5, 6, and 7 nights after the full moon on August the 30th, and we'll be, around 10 o'clock, between 10 and 11 o'clock at night, in that 3 hours after sunset window, and we'll be doing those 3 nights and documenting, as much as we see. But we take paying customers out, so it's a little bit hard to experiment and look for the ones that aren't as common or as easily as frequently seen. So we'll be taking out 3 different boats to 3 different dive sites in Grand Cayman, on the east end of Grand Cayman, and I'd love to. I haven't seen any of the branching corals for about 5 years now, but we're slightly off on the timing. But mostly the different boulder corals, and seems to be the most reliable for us that we monitor. Hadn't thought about collecting, but that's certainly interesting too.
Well, don't disparage your efforts, Steve, because I believe you probably have the only information that exists for Grand Cayman. So it's great that you've started already with the tourist dives.
Yeah, I
And,
mean, yeah.
Go ahead, Steve.
There we've been since 2003. We only missed 2008, but we've done it every year for 18 years now, so quite reliably. But I would like to try some different nights, and it's fascinating to hear about what's happening in June in broad daylight, really. So certainly for next June, that's something I'm going to try. And maybe I'll try and get out in July, but I might on the 11th, 10th, and 11th nights. So that certainly would be interesting. A lot of the DLABs, we have had a lot of damage from Skittle D, so it is some of these species will be hard to find. But the branching corals are quite plentiful, and but the ones the most susceptible, Skittle D, are sort of dwindling in numbers. So we'll certainly be looking out for those too.
And I saw a few other people had posted in the chat that they'll be going out as well. It's great to see so many people going out. Christine, I see in Grenada, Valeria, the Bahamas and Colombia, Gabby, your groups in Roatán, LZ, I saw that, and Tatiana, you're in Aruba. That's great. Hopefully you can get out as well. Anya also posted a video, and there's instruction manual with guidelines that she posted some really good links there.
And Lisa Karn in Belize will be going out, possibly for DLAB. And so this is great. And you're getting converts.
And I think I'll real quickly show everybody, if you wanted to, if you can see my screen.
Good. I was about to ask you.
Yes. Can everybody see my screen?
Yes.
Okay. Similar to the collaborative effort that we did with the Caribbean Cooperation Team and many partners throughout the Caribbean to track coral disease outbreak with the stony coral tissue loss disease, and also more recently last year with the diadema die-off, we have been working with Anya on bringing information together, and this is kind of an open invitation for everybody to help collaborate on this. All the work we do is collaborative. And we put together the Caribbean coral spawning database, and we're collecting the information on here. It's basically shows you just some basic information. Anya's previous talk that she did on monitoring coral spawning and the one she did today will be available here. We also developed a spawning tracking map, which I will show you in just a minute. But on here, the map will show places where when you enter the data, it gets posted immediately. This is data from previous that Anya had shared. So we're going to start a new map for 2023 that will start putting in the information from DLAB from the different areas. The video that Anya showed is available here, and all of the resources that Anya has been sharing from all of the many amazing partners in the Caribbean who've been putting together spawning calendars for the Mexican Caribbean, Dominican Republic with Fundamar, Southern Caribbean with Carmabi, and Perry Institute of Marine Sciences, their new prediction calendar as well as the spawning template data. If you're entering a lot of detailed data, some people collect detailed data, some collect more basic data. So you have the ability to do that. There's also links here. If you're doing coral spawning work and you have a link that you would like to share with us, we'll be happy to add you on here. And then those who are sharing the data, we would like to share and get your information out there. So you're welcome to share your logo as well. So we invite you to share resources, and we'll, similar to the stony coral tissue loss disease, we'll keep this updated as information comes out. I did want to show you the spawning report, if you click on that little dot, it brings up, this is an ArcGIS based survey form. It's nice and easy. We base this on the form that Anya was talking about, that Karamavi had originally put together in the Excel form, and we put it in a simple tracking form. So just real easily, you can just enter your name, email, the date that you did the observation, and basically just, did you observe coral spawning? Once you click this, there's a series of questions. You keep going. What country of survey? We ask this because it does help us organize the data better and on the map. And you can put your lat long in. The lat long is what helps put that dot on the map, so we know where you are located and where your observations are located. You can put your site name. If you're working at a specific depth, you can give that information. And if you have an area that you're monitoring, let's say you're just doing a 10 by 10 meter area that you're monitoring, you can put in the approximate area of your site monitored. And as Anya mentioned, the start and end time of the spawning is really important, so you can put that information in as well. And the date of the full moon, you can put that in. You have a list here that you can click which species. You can say no species. As Anya said, even if you go out and you don't see anything spawning, that data of no data is really, really important as well. But let's just say you saw the Cervicornis and Palmata. Once you click those, it gives you the opportunity to put in the number of colonies observed and the colonies that spawned. As Anya said, like in Cuba, they saw a few colonies out of the many that they were monitoring, but still that's really good to keep track of the number so that the percent can be calculated. And then if you did see other species that did not spawn, you can go ahead and collect those. Again, you know, saying, no, we didn't see anything else like any of the brain corals or any of the other corals. And it has a whole list here. You can just go through and select. You can add any comments. So for example, if you had questions or if you had additional information you want to put in, you can add it here. And best of all, and this is what we found so helpful when we did the stony coral tissue loss disease and the diadema die-off, was the ability to upload pictures to confirm, you know, is this spawning? You know, if you don't actually see it, but you see that it might be setting, you can upload your photos. And these photos have been so, you know, helpful in tracking. And then you can submit your form. And again, all of that information gets put on the tracking map so that we, you know, everyone can see where you are located. So we're hoping that this is a good resource for everyone to use. And I'll be happy if anyone has any questions or wants to know how to use it, or if you have information you'd like to share on the page, on the coral spawning page, or videos, or just examples of, you know, what it looks like when DLAB actually starts to spawn, you know, feel free to, you know, reach out to myself or to Anya, and we'll be happy to move forward with that. So if no one had any other questions, I'll stop sharing there.
Well, let's ask and see if anybody has any questions before moving on.
Oh, okay.
Are there any questions about the new feature on the AGRRA website that Anya and Patricia have put together, along with the help of some of our other colleagues at AGRRA? Please use it, because everyone will benefit from the collective information that we acquire, as Anya has said earlier. And if not, thank you, Anya. Once again, you have helped energize our folks, some of whom are feeling pretty, I would say, jaded and weary after 3 or 4 years of dealing with dying corals. It is so energizing to start thinking about making new life and starting to repair the damage that's been done. And more along the vein of larval propagation, I will now ask Gabriela Ochoa, the RMP, and she from RMP and also MPA Connect, who will now overview last month's larval propagation training workshop in Roatán. And can you start sharing your screen, Gabby? And after she speaks, I hope we'll also hear from at least one or maybe 2 of the attendees who are also on the call today. So here we are. Okay. You're on mute.
Am I sharing the right one or are you looking at my
It's
notes?
the wrong one. It's the wrong
Yeah.
one.
Okay.
Yeah.
Let me see how. One more chance here. Let's see. What about now?
Yes.
Yes, perfect. Awesome. Well, thank you all so much for this opportunity. I'm really happy to see some familiar faces, some which were actually in this Coral Rescue Exchange last month. So as Judy mentioned, we hosted a Coral Rescue Exchange in Roatán, focusing on coral spawning monitoring and assisted sexual fertilization and rearing of species susceptible to stony coral tissue loss. For this Coral Rescue Exchange, we focused on Diporia labyrinthiformis or DLAB group rain coral. And this exchange was actually hosted by the Roatán Marine Park. And we had 4 members of our network from Barbados, Dominica, and Grenada. And we also were super lucky to count with Eduardo Avila, who's a roving technician from Sea Corps. From Barbados, we had the Coastal Zone Management Unit. We had a representative from Superior Scott Marine Reserve, Simon, who's in this call as well. Shanae from Barbados, who's in this call as well. And we also had 2 dive shop owners, Christine Finney, who will speak a little bit more about this exchange as well, from Eco Dive Grenada, and also from Barbados, Barbados Blue. Awesome. And the reason we had this kind of a multidisciplinary group is because we wanted to highlight, these collaborations that happen when we're doing this work. And the Roatán Marine Park has been really successful at working with dive operators through stony coral tissue loss disease interventions, and now moving forward to working with dive operators through their coral spawning work. So every month, the Roatán Marine Park works with a different operator who gives boat time, fuel, and captain to be able to conduct this work. And move to my next slide. And so during the Coral Rescue Exchange, we focused on kind of like the dry activities, which are, getting ready to set up your area where you're going to be doing this assisted fertilization, setting up the cribs, and also, conditioning and cleaning your substrates and getting all that ready outside the water, in the water. Also, we worked with our partners to run them through how they would set up a coral spawning monitoring, going through different predictions from different calendars and getting them started in their own calendars. Simon will share a little bit about how, through what he learned through the previous meeting, he was able to get out there in Dominica and actually observe spawning for DLAB. We learned, as Dr. Anastasia shared with us, how to build the nets and how to deploy them properly, and how to do the collection of the gametes. And of course, we got to observe some coral spawning, which is also really hopeful and beautiful. And one of the highlights also of our training was to run our partners through what we call a coral rescue Canva, which runs partners through different questions that they should be asking themselves when they're getting ready to deploy different coral rescue approaches. In this case, of course, we were focusing on coral spawning monitoring and assisted fertilization. This tool is actually available on our website, so you're able to download it and run through them. And I'm sure some of our partners will share with us how they found this tool. So yes, this was basically our Coral Rescue Exchange in Roatán. This was a follow-up activity from our 9th peer-to-peer exchange, which we hosted in Key Largo. And many of those that participated reported that they wanted to get more experience on this type of work. So I wanted to thank everyone who made this work possible, especially the Roatán Marine Park for hosting us for a whole week, and everybody else who participated. So thank you so much.
Well, thank you, Gabby. That was a wonderful introduction. And I have to tell you that Simon has already had to leave. I'm not quite sure what his emergency is in Dominica. But Christine Finney has graciously offered to fill in for him and also give us what she thinks he might have said, and also give us her impressions of that workshop. And she's smiling, so you can tell she had a fabulous experience. So Christine.
Good afternoon, everybody. Can you hear me?
Welcome. Yes, we can hear you.
Thank you. So I honestly have no idea what Simon would have said, except save more corals. So that's for Simon. The rest of it, I have to say, Gabby, close your ears, but the week Gabby organized with Christine and MK Connect and Noah and everybody else was amazing. I had probably the best week doing choral stuff I've had since before COVID. And for that motivation, if nothing else, I am forever going to be grateful. It was really good to see something positive with hands-on attainable solutions as opposed to just watching Skittledy kill everything. So I don't know. I mean, I'm really excited for Grenada. We're trying to get out this week to get some DLAB. I know it's not June, it's July, but we're still hoping for the best. I would love to follow up with C-Core more if people haven't seen that layout and design. I was super impressed with the functionality and the purpose and what we could achieve in a couple of days. And I just think that the team was amazing and the experience was really good. And I'm very grateful to everybody. And Shawnee and Ajani and Simon were great company. I don't know, it was a really good week and really good to see some positive, as Judy said earlier, instead of just things dying, it was really good to see stuff with potential. And it makes a lot of sense. I have to say in the last 2 weeks though, I've been trying to pick a dive site for our DLAB spawning, and it's depressing because we've lost a lot of colonies. So trying to find an area that has enough mature sized colonies in a decent viz range and stuff is interesting on its own, but it's part of the process and we're into it, so we're on it. But I just want a big thank you to everybody.
Thank you so much, Christine. And since Simon's not here, I do see Shawnee there. I'm not sure if she wants to share some words, but on behalf of Simon, I actually just wanted to commend him because Simon was actually during the Coral Rescue Exchange, working and monitoring with us, and simultaneously also folks in Dominica were also monitoring. Unfortunately, they didn't see as many coral spawn, but they're gonna get ready to do it this month as well. And I know that the experience that he received from the training has been really helpful to put everything into practice. So I just wanted to add that on behalf of Simon.
I have a question for Gabby and anybody else. If I have the resources for 2 days this week to get out and look for DLABs, it looks to be like the 13th and 14th are my 2 best days.
Absolutely.
Is that what I'm aiming for?
Absolutely. Those are the days I would go out, definitely.
Okay. Do we have anybody from Trinidad on this call? Because I remember Simon saying when we were in Honduras that Trinidad got their spawning like 75 minutes before sunset. So I was just wondering, they're the closest island to us from anybody that I've heard reported. So I was just curious if anybody knew if that was real or I misheard or Simon misunderstood or no?
We'll have to ask him.
Not sure, but it's possible. And again, this is sort of another reason to push the idea of having the spawning reporting done by people so that we don't have these doubts and we can map these times and the days. It would just make everybody's life a little easier.
Very good.
Thanks.
Thank you.
Yeah, and I'm not sure, Christine, if any of your sites, as you saw, our site in Roatán is pretty much snorkelable. So if it reduces cost on you to go out and snorkeling, you might be able to observe it if they're shallow enough. So
yeah, no, for us it'll be the boat support that will be the cost mostly, which is internal because it's what we're up to. But also our visibility, we're too close to South America. Our visibility is not always as bright and clear as Roatán. So you'd kind of want to make sure you could get up and personal with the coral because from a snorkel perspective only. We get some green emerald at this time of year, so it's hard for us necessarily to see the bottom, even on our 6-7 meter sites. But I like the suggestion.
Completely understand. Any other questions about the Coral Rescue Exchange?
Well, I will add something that I would suspect Simon might have said were he to be on the call, because when he returned from Roatán and I was talking to him, he was so amazed, overwhelmed, appreciative of what a wonderful job the Roatán Marine Park is doing in terms of its positive advertising of their activities, its recruitment of volunteer divers to go do things with them, its successful fundraising activities. its ability to operate lots of programs external to having to rely on government support. And I think he felt he'd learned as much about how to operate a successful rescue mission as he had specifically had any learned details about collecting spawn. And I don't know if Shawnee or Christine would care to comment on any of the aspects of how the park in general is organized.
But I mean, I was floored. I don't know who I just cut off. I'm sorry.
It's okay,
but-
Christine.
Hey, Shawnee, you get to go
Hi.
next, and you get to say lots. No, the Roatán Marine Park. I mean, I was kind of floored because from our institutional structure down here in Grenada, having a non-governmental unit that first of all could be called Marine Park and then has such functionality. And I know they've of course had challenges and had a rough road coming up, but what they've accomplished since 2005, I think, is trophy worthy. So they ran an amazing week, and the personalities they have inside, what they accomplish, how they accomplish it, and their stakeholder involvement is impressive. Goals, it's good to have goals. Shawnee, you're up.
Hi, thank you, Christine. Well, I guess I'll just speak on it generally. I too had a great experience. Everyone who was involved, I think the company made a lot of the experience like miles better. And I too was very impressed with everything. I think I went in as assuming that doing any type of sexual, I guess, like fertilization would require these vast amounts of resources, and you wouldn't necessarily be able to get a lot done. But being there and recognizing it's if you can get people in the water, you can actually do way more than I would have expected beforehand. And speaking on, I guess, just the general way and how the governance structure and the culture, I guess, of Honduras, and how much, like you said, they're able to achieve, everything kind of just fits so well together. It's I think a framework and structure that I would love to see implemented in other places. So it was just a magnificent experience generally, to be honest.
And Gabby, I heard that you were wonderful at arranging the logistics.
Thank you. Thank you so much.
Somebody told me you're a rock star. Now, I'm surprised you didn't mention the cribs.
I did. We did assemble. Maybe I didn't spend too much time, but everybody got experience setting them up.
Yeah, let me ask you how it went, how they're holding up now after a couple of weeks. Because
So our crib is still holding up. It's for those of you that are not very familiar with Roatán. I mean, it's located in Half Moon Bay, which is right in front of the RMP's office. So it's kind of like a short snorkel. We were a bit concerned because there's a lot of tourists, right? That, I was telling our team that putting a sign to not come in is like an invitation to come in, People are like, oh, I want to see why I can't go in there. So we had a lot of people, climbing into it. Some shackles got stolen from it. So it's still holding on. I'm happy to report that we have recruited onto the substrate. So we do have, little baby corals growing strong. We're waiting on them to get a bit stronger to finally outplant them onto the reef. But yeah, this was a really great experience for us, and I'll speak from the Roatán Marine Parks experience too, of hosting groups. I know that a lot of folks in Mexico and Dominican Republic host groups constantly, all the time, every month, right? And it involves a lot of work and logistics. So it was good. And as Shawnee said, the company made it even better because we had a group of experienced divers. Everybody was comfortable in the water. Everything went really, really smooth. So we were happy to host you all, and that you saw that it can be easy, not always easy, but if you can get people in the water, it doesn't take a lot of things, to get it done. And you don't have to, something that Simon learned is you don't have to monitor, 40 corals, and you really don't need that many corals to fertilize them, right? You just need a few. So I think getting this real world experience was really helpful. but the crib is holding on.
Oh, I'm so glad to hear. And I'll just say hello to Hannah Lockham, who is now joined in and said that they observed DLAB spawning in Tobago 11 days after the full moon in both May and June this year. So there, we do have another data point already. So that's wonderful. And now I would like people to write in the chat if any of you are interested in more training, either in terms of larval propagation, like was offered last month in Roatán, or in terms of asexual fragmentation and grow out of colonies, larger colonies, such as Stacy Williams was talking about earlier this spring, and so was Simon, of course. Or if you would like to have information about aquaculture of herbivorous diadema to facilitate in the preparation of the substrate to be suitable for corals to live and grow, please write in the chat who you are. Well, we'll know who you are, but where you are and what kind of training you would be interested in, because we're always on the lookout for more opportunities, and we need to, and it's good to know who is interested and would like more training. And while you're doing that, I am going to share my screen, I hope. And we will generally share our concerns for the current warming trend in the Caribbean. And after I talk about that for a little, Patricia Kramer will come back and talk about AGRRA's freely available Bleach Watch underwater data sheets and online reporting tool. So let's see if we can get started on this last part of the program today. Oh, come on, share. And can I do this? And oh dear. So I have those unfortunate things at the bottom too, but I'll try to ignore them because I don't know how to get rid of them. The reason we're so concerned is that here's the problem, by mid-June, unusually warm atmospheric and seawater temperatures were recorded, and they were widely politicized around the world with maps that look something like this, and those dark red lines show the temperature this year in this June just escalating incredibly, almost off scale, you could say. And people in many parts of the Caribbean started to notice that the water temperature was getting warm. Fragments of Hope's Lisa Karn, who was on the call today, had been alert to the fact that there was some paling bleaching in Belize since the 10th of June, so she was already worried. And the first actual real bleaching of the elkhorn corals was seen in the southern Belize, in the lagoon in southern Belize on the 27th of June, when she had these photographs taken and sent to me. And on the next slide, we can look at the NOAA's degree heating watch map for the 28th of June. So that's the following day. And degree heating weeks are simply defined as the accumulated heat stress above local bleaching threshold during the previous 12 weeks, with a score that runs, as you can see, from 0, pale blue, to black 10, and with the reds that you can see along the coastline of Belize and around parts of the Yucatan Peninsula and down parts of Honduras and Nicaragua, and even offshore in and even, I'm sorry, off the coast of northern Panama, being seriously heat stressed already. And so that's not good. That same day, this is only, I think, 12 days ago, a graduate student from SSU called Shani Ashul arrived in Bocas del Toro that red dot off of Panama. And the following day, she jumped in the water, and at her very first dive site, she discovered that all the common corals, the finger corals, the lettuce corals, the fire corals, were already extensively bleached. In other words, the bleaching was well advanced by then. And even some of the massive SIDs were getting pale or were totally bleached. A few days later, on the 3rd of July, you can see tendrils of warm water were now extending further out from the coastline of the Western Caribbean into the open Caribbean Sea, and they were joining up together to form sort of like a ring of warm water in the Gulf of Mexico. And that spot that she had observed the day before down in Panama was still hot. Now, last Friday, which is only 4 days ago, Lisa Karn received these photographs from off the barrier reef in northern Belize, and both the elkhorn and the staghorn corals at this point, as you can also see, are extensively bleached. And if this keeps up at this current rate, some of these creatures unfortunately are going to die. And the degree heating week map for that day, last Friday, where the northern Belize site is shown at the red arrow, you can see that it's still hot. The warming in the Caribbean Sea is now, covering an ever greater area. The Gulf of Mexico is increasingly becoming impacted. And the warming now wraps around Western Bahamas, Northern Cuba, is getting close to the Hispaniola and Puerto Rico, and is even extending up along the east coast of the United States. So with that grim news, I'm going to pass you over to Patricia, who will tell you what we can do to at least document for posterity what's happening or what is going to happen this summer. So Patricia, I will stop screen sharing.
Hi, Judy, thank you. So as I think many people who have been tracking stony coral tissue loss disease are familiar with our reporting tool for stony coral tissue loss disease, integrated in that is the ability to put in bleaching observations. If you go to the AGRRA website and you go to coral reef monitoring, first, the coral spawning webpage that I talked about is available. It is no longer password protected, so you can access it. The one thing with the coral spawning, we will be updating the map, so that's 2023 data. For coral bleaching, you can select coral bleaching and you can go down. There are two different survey forms, a basic one, which is basically just looking at, if you just have information on the species, but you don't necessarily do quantitative counts, you can go down, enter all of your basic site information, select corals with bleaching, or if they also have SCTLD, you can select both. And then there's a very simple checklist. You can do a checklist of which species that you saw bleaching. Like if Lisa was entering data, she could, which she does a lot of, she could just select, how many colonies she saw there. This is the basic form. We did work with the Healthy Reefs Initiative to make a detailed form, which is this one here. And there's a video here of how to enter the data and also a data sheet available. But if you click on the advanced data sheet, you can go in and you can select the species and put in more quantitative data. So this is the quantitative data. Again, you can put in your, putting in your location is the most important because that's what pins the information to the map. And again, this is a live tracking map. And it helps us track real-time data, which is really great to be able to do real-time instead of looking at it in the past. You can go down in here. If you did see corals with bleaching, you could select it, or if you saw it with SCTLD. And once you select the information, let's say you saw some cervicornis and palmata bleaching, you could go down and it allows you to enter in the quantitative data. So for example, you would select the ACER. We use a 4-letter codes, which is the 4-letter code is the first initial of the genus and the second, the first three letters of the species. But here you can put in how many colonies you saw of healthy, fully bleached, partially bleached, or pale. And this is helps us to do tracking of quantitative data. So we have both forms available. I went through this very quickly just for the sake of time, but again, there's a video to show you how to put in the data. But the main difference between the basic and the detailed is the quantitative data. And again, we worked with the Healthy Reefs Initiative to help put in this quantitative data. And just to kind of show you how it works, we just put in, actually with Lynette Roth, who is our database manager, this morning, we took out and separated out just 2023 data. So you can put that in and you'll see just the data. You do have the ability to go in, if people did do like total bleaching or fully bleached, it will show that data. You can see that it comes up separately as separate dots. And then we also have the archive data in our previous map. And if we zoom in here to the MAR, the Mesoamerican Reef region has a coordinated coral bleach watch program with the 4 countries of Mexico, Belize, Guatemala, and Honduras. And every year they go and they track data. And as you can see, I think most of these sites are from Fragments of Hope, but you can click on a dot and it gives you a pop-up table and it will give you the data. And it gives you what did you observe, if a healthy, bleaching, disease. And if there's photos, you can upload the photos as well. So these are some new updates to the coral bleaching part, but we did separate out the data specifically for 2023 so we can see what's happening here. So again, we welcome you to share your data. If you have information, educational information you want to share with other folks, please, reach out to us and let us know so that we can share information. If you have great photos, you can add that as well. So again, we welcome you to share the information and enter your data and help us track. As Judy said, this year is predicted to be, high sea surface temperatures. We're already seeing it in some areas. And I think this is a really interesting opportunity, as I think Christine said earlier, many of the reefs have already been affected by stony coral tissue loss disease. Some may be affected by bleaching. Our key question that we have is, with the corals that are still remaining, are they able to reproduce and spawn? So that will help us. So if there's any questions, I see Lisa, raise your hand, go ahead and ask your question, Lisa.
Hi, thanks for that overview. Hi, everybody. Lisa Karn in Belize. For years, we've been using an Excel data form, a data entry form that was created originally, I believe, by Melanie McField, HRI, for the rapid assessment, what she calls a swim bar drop or something like that. So we can share that link. And I was going to say that for ease of entering it into the AGRRA way, there's some formulas in the Excel sheet where it counts how many of each species. So I think that might be the easiest way to have someone entering in from the Excel sheet to the AGRRA database. Because we've collected a bunch of survey data that we need to share with AGRRA. And I think that would be the easiest way to do it based on just now seeing how you do it on the AGRRA site. And so to answer Gabby's question, we also have a link we could share. We did a kind of refresher video last year that was not intended for first time people doing the bleaching surveys, but more of a refresher for people that, only maybe do it once a year. So I can share that. I was unable to update it quickly like I wanted to with this year's alert. So last year's video has last year's alert information, but it walks you through the protocol of how to do it, including the raw data forms, and also the Excel form that we then use to enter the data from our raw data sheet. So there's unfortunately 3 steps. So we have the form or the blank sheet that you use in the field. Then we have the Excel form that we use here, that we've been using here for over a decade, and now the AGRRA data form as well.
Yeah, and there are other ways. I mean, the beauty of this form is that it's not dependent on any given method of collecting the data, and that is one tried and true method in Belize. But people could also just do roving surveys, as long as they made sure they didn't pass over the same part of the reef more than once, and count everything, every coral that they see within a species that's bleaching susceptible. And or they could do fixed transects, you know, with a transect line or a meter tape. And those surveys in a defined spatial area will allow you to calculate, you know, density, and get density information, which is, you know, a lot more quantitative and rigorous, if you like, but it takes longer. So it's a trade-off. If you want to just get an idea over a large spatial area or along at many sites along a reef tract, it's better to do something that doesn't require fixed transects. And roving diver or the method with the bar that Lisa's talking about, you know, they're all available, but you end up with numbers, and you can write the numbers in the online data entry tool, and there'll be a place where you can say how you collected the data.
And just to expand on that, the usefulness of these tracking tools has been tremendous. On these stony coral tissue loss disease, this was such a great communication tool and tracking tool to work with over 35 different countries and territories throughout the Caribbean. This is the stony coral tissue loss disease map, and we had over, I think, 2,000 surveys that have been entered. So it's just this, as Anya always says, it takes a village. And I think this really kind of brings home that point. And the same with diadema, when we worked with the diadema network, response network, you know, being able to track within just a few months how quickly diadema had died off. So just encourage that the collaborative nature of everything so that we can, you know, continue to work as a village, as Anya says.
Yeah, I think the diadema response network tool, when the diadema were dying in so many different areas and so quickly last spring, is probably a better model for what we might very well end up in the summer when the corals start bleaching. If the continued warming trend doesn't get reset sometime soon, which doesn't look likely, and the El Nino event that is, you know, developing this year hasn't even really kicked in our part of the world yet. We could very, very, very quickly have corals bleaching all over the place. And, you know, when that happens, it would be so useful to be able to present a Caribbean-wide perspective, overview of what is going on. If for no other reason than to present it to, you know, the major developed countries and say, look what you've done to us now.
I'm just quickly showing just the diadema health
Yes, thank
tracking
you.
map.
Most of
Judy
that,
was
yeah.
mentioning.
Most of that was filled in over a period of 4 months.
Yes. Yeah, so I think it was over 700 reports. And what was really great is that it's scientists, managers, citizen scientists, you know, so many different audiences involved in helping. And this one actually led to the collective effort to eventually identify the cause of what was causing the diadema to die off. So there is great usefulness in sharing information. Okay, Judy, back to you. I think, I didn't know if anyone, I think, have any other questions on that. Judy, did you have other questions or?
I don't have any other questions. I don't know if anybody else does. I don't see any raised hands.
Lisa just raised a hand and we had a
Oh, Lisa.
question. Yeah.
Lisa, are you asking another question or did you forget to?
I am asking another question. I'm sorry if you guys covered it already.
No, that's
Do we
okay.
have any information on hotter temperatures affecting coral spawning times or regularity or any information in general? I know there's a lot of information about sea temperatures affecting fish spawning patterns. Is there something already out there for the coral spawning patterns and temperature?
Lisa, there are a couple of publications, and spawning can be stalled, or reproduction can be stalled if the temperature is too hot. And sometimes they will spawn if they're developed enough, but then the next year or even 2 or 3 years, spawning could be affected.
Thank you.
You're welcome. I'll try to get the papers to you if you want.
If you give them to us, we'll put them online as well, if we can.
Okay.
And how sad it would be if people are now learning what months, what days of which month spawning occurs, and next year and the year after it's been curtailed because of what else is happening this summer.
Yeah, it actually brings up a good point that for species that tend to bleach more readily, let's say like the orbicellas, it might be good in your surveys to sort of knowing which colonies have bleached and which haven't, so that if you're following reproduction or coral spawning in future years, you know which ones didn't bleach and are more likely to spawn.
Oh, that's very important. Yes. Okay. Good point. Does anybody else have any other questions or comments? If not, it's the magic 1:30 p.m. So we are on schedule for a change. And thank you all so very much for joining in today. I hope you've learned a lot. And Christophe has just asked a question. Maybe we'll answer you afterwards, Christophe, because it looks pretty long. And we'll close this meeting now and thank everybody for attending. And you can look for some more materials on the website as we get them. And good luck to everyone who is conducting any kind of spawning monitoring between now and September, when we will have our next meeting.
And thank you to Anya and to Gabby and others for presenting today. We really appreciate sharing your great wealth of information.
Yes,
very
it's really
welcome.
wonderful. It
Yes,
gives me hope. So I like the positive side that you're already, out there looking for spawning corals, and also starting to, Gabby, grow baby corals, which is exciting. So look forward to hearing more from everybody as they continue with the coral spawning.
And
Your
thanks.
gamete bundles rise to the surface.
May lots of gamete bundles rise to the surface. Amen. And thanks also to Christine and Shawnee for their comments on the training. We appreciated that as well.
Bye, everyone. Take care.
Thank you. Have a wonderful week.